Hello everyone, myself Dr. Yogi Pandya, Assistant Professor at LJ Institute of Pharmacy at LJK University. So today we are going to study about gravimetric analysis. So as we know from the name itself suggests that gravi means to weigh and metry means to measure, right? So it is a quantitative method for measurement of the weight of the sample, right? So in this, we are converting the main analyte into some known composition that can be separated, right, from your sample and which can be accurately weighed, right? So it is a quantitative analytical method in which mass of an analyte or its derivative like precipitates or crystals or co-precipitates which can be accurately measured, right? And it is a branch of the analytical chemistry in which quantity of analyte is being analyzed on the basis of the mass of the solid, right? So what is the main principle behind the gravimetric analysis? As we have discussed that the principle behind gravimetry is the mass of the ion in its pure compound can be measured accurately, right? And later on, the mass percent of the same ion can be calculated from the known quantity of impure compound, right? Here you can see the assembly of the Buckner funnel and the flask is shown, right? You can filter out your sample, which is connected with a vacuum pump, right? Vacuum source. So later on, you can take out the sample from this Buckner funnel assembly and you can dry the precipitates and then accurately you can weigh the precipitates, right? So now what are the main key features, right, of this gravimetry analysis? First of all, we can see it is a direct mass measurement technique, right? Directly we are weighing the sample, so it makes highly accurate method. Secondly, it is a classical technique, means conventional technique. It has been used in analytical chemistry since many years. Third one, you can see minimum instrumentation is required, right? We know that in sophisticated analytical methods, we require very costly instruments, but in this gravimetry, we require very much less amount of tools or instrument like simply balance, crucible, filter paper, right? Drying oven, furnace. Only similar laboratory tools are required. So it is also low operating cost and we are also using less expensive reagents as compared to the conventional technique and minimal equipment requirements are there, right? So it is a feasible technique in analysis. Even it can be also used in macro and micro scales, right? Means small quantities as well as large quantity of samples can also be analyzed, right? So there is a benefit of this gravimetric method. Now we'll see what are the general steps, right? What are the steps in the gravimetric analysis? Initially, what we are doing, we are weighing the sample. Sample which is containing your analyte that is accurately weighed, right? Then we are dissolving this sample in some suitable solvents like water, acids or base, right? Or any other organic or inorganic solvents. We are dissolving the sample. Then we are adding some reagent so that main analyte is converted into some precipitate, right? Which is insoluble precipitate. This step is important, right? Third step, that is precipitation, in which by addition of the reagent, selectively your analyte will be precipitated. Then later on, we are doing filtration by using filter paper or other filter assembly like Buckner funnel flask assembly we can use. Then next step, washing of the precipitate either with solvent, right, in which the soluble impurities can be removed. Then another step is the ignition or drying, in which we are heating at particular temperature so that volatile solvents, right, water or other any volatile impurities are evaporated and your sample is completely dried, right? And in the last step, the dried precipitates are cooled, right? And accurately, they are weighed to analyze how much analyte is present in your original sample, right? So these are the general steps which we are carrying out in the gravimetric analysis, right? Now, what are the types of gravimetric analysis? In this, mainly we are discussing here three types. First is the precipitation, second is volatilization, and third is the crystallization. So as we know, in precipitation, we are converting your main analyte from the sample into precipitates. For example, barium sulfate for sulfate analysis, in which the sulfate ions are being converted into BSO4 precipitates, and then it is filtered, washed, dried, and weighed, right? So those analytes which can be converted into precipitate form, in that we are using this mainly the first technique. Then in second technique, that is volatilization method. In this, we are trying to remove the volatile impurities. The loss of the mass is calculated from the analyte's concentration. It means how much mass is lost by ignition or by heating at high temperature. That is determined, right? Means the organic content has been determined from the loss of the mass during ignition, right? And the third technique is crystallization, in which we are converting your analyte into the form of crystals. And mostly this technique we are using when we require high purity of chemical analysis. At that time, we are doing this crystallization method, right? Then how you can produce precipitates, right? What are the methods of precipitation? For example, you can use some reagents like silver nitrate, right, which can react with chloride ions to form silver chloride, AgCl, sparingly soluble precipitates, right? Similarly, you can precipitate by using pH, by changing the pH, right? For example, barium sulfate precipitation occurs at in acidic pH, right? So by pH adjustment also you can convert your analyte into precipitates. Thirdly, by either using temperature, right? The formation of precipitate may be hindered or either be enhanced by change in the temperature, right? Means certain precipitates might be soluble at high temperature, some might be soluble at low temperature. So based on that, you have to adjust the temperature so that most of the salts or most of the analytes are completely converted into precipitates, right? So controlling temperature is also important during this gravimetric analysis, right? Here is the assembly. You can see filtration of crystals or precipitate by Buckner funnel flask assembly. Here is your mother liquor, means your sample plus reagents, right? And here is the filter assembly, which is kept over here connected with a vacuum pump, right? So that you can take out the air and you can filter out the individual precipitates. Now in volatilization, as we discussed, we have to heat at particular temperature so that the volatile impurities are evaporated. For that, we can use this instrument. Now, what are the important factors in gravimetric analysis? Right, we'll discuss that. First of all, purity of precipitate is most important, right? During analysis, what happens? Along with your main analyte, some other impurities or some derivatives or byproducts, they also form precipitate, right? So that causes error in the analysis, right? So that is most important point, right? Precipitate must be pure to get accurate results, right? And the contamination or the co-precipitation must be accurately handled during analysis. For example, you can see chloride contamination in sulfate precipitation can lead to erroneous results, right? So for that, you have to remove first those impurities and then you have to go for the analysis, right? Second is complete precipitation. Sometimes what happens that your main analyte is not 100% converted into precipitate. So for that, you have to make sure that your analyte fully reacts and it is converted into particular precipitates. Otherwise, the incomplete reaction will make error in the analysis. And these unreacted species can lead to erroneous results, right? Third point, we can see the filter paper or the membrane quality which you are using must be of the extra pure grade. Means it must not allow the fine particles to come out, right, during the analysis. Otherwise, the precipitates which you are weighing must not be 100%ly, result might not be achieved, right? Then some commonly used reagents in gravimetric analysis, like you can see here, silver nitrate, most commonly used to precipitate the chloride ions which is present in the sample in the form of white colored insoluble AgCl precipitates. Secondly, you can see barium chloride reagent, mostly used to precipitate sulfate ion into barium sulfate, right? Third is the ammonium hydroxide. Mostly it is used to adjust the pH and also helps in the precipitating of the hydroxide ions, right? And fourth, we can see potassium chromate, K2CrO4, that is used selectively for precipitating the lead metal in the form of lead chromate, right? Then some examples of gravimetric analysis. First, we already discussed that chloride ions can be mostly present in the water, so they can be precipitated out in the form of AgCl, right? And that can be analyzed. Then easily by reacting with the reagent, it will form insoluble silver chloride precipitate. Later on, it is filtered out, right? Filtered and washed with the suitable solvents to remove the contaminants. Then the precipitate are thoroughly dried, right? And the weight is done accurately. And from that basis, you can calculate how much mass percent of the silver as well as the chloride ion have been precipitated together and how much amount of chloride ion was present in the original sample of the water, right? Now we'll discuss what are the main advantages of gravimetric analysis. First of all, it is accurate and precise method because there is direct mass measurement of the precipitates, right? Your analyte is directly converted into relative precipitate and you are accurately weighing those, right? So it is simple technique and accurate method. Secondly, as we have discussed previously, that minimal instrumentation is required, means can be easily done with our common lab equipments like balances, crucibles, filtration setups, etc. Then cost effective means less expensive method as compared to instrumental techniques, right? Even the cost of reagents are also very less as compared to other techniques of analysis. Then high sensitivity means even in the lower concentration also we can detect the mass, right, of the particular precipitates. So as well as also it is a sensitive method. Then what are the drawbacks, or you can say what are the limitations of this gravimetric analysis? First of all, it is a time consuming method, right? Because so many multiple steps we have to undergo, right? Precipitation, filtering, washing, drying. So overall, it is a very slow process, right? Then even sample size, even more suitable for large amount, but not ideal for a lower concentration, like in parts per million or parts per billion. It is not a suitable method. But when your amount of sample is in large amount, right, like in grams, then we can use this technique. For milligram, micrograms, this technique is less efficient. Third drawback, you can see interferences, right? We know that impurity which are present along with the analyte, right? They will also co-precipitate and they will lead to some error-based results, right? For example, chloride ions, when determining sulfate concentration, it can lead to co-precipitation, right? So that is also interference during analysis, right? And what are the applications of gravimetric analysis where it is used? Mostly we are using them in the pharmaceutical industries for determination of active ingredients, for excipients, drug formulations, right? We are using this technique. Even for the environmental testing, we are using this gravimetric analysis like for heavy metal analysis, lead, mercury, anions, sulfates, nitrates. Even in the water or soil analysis also, we are using this method. Even in food quality control also, we are using this gravimetric analytical technique. Even in metallurgical process, we are using to analyze ores, refining process, iron determination, copper determination. Also, we are using this gravimetric method of analysis. Then another example like assay of drug substances. For example, assay of sulfate ions, we are doing in the form of barium sulfate, in assay of chloride ions in the form of AgCl, right? Then purity testing also, we are using this technique like raw materials, excipients, salts, right? As per the pharmacopoeial specification. Even indirectly in the limit test also, we are doing this gravimetry analysis, right? To identify how much amount of heavy metals or other impurities are present in your main pure sample or either excipients, right? Then moisture content analysis, also loss on drying, how much amount of water is evaporated. And again, you weigh the sample. From that also, you can determine moisture content. Then residue analysis, that is sulfated ash, residue on ignition, right? Loss on drying, loss on ignition. That is also one of the gravimetric technique of analysis. Then quality control of finished product, right? That also we are using this method. Environmental and waste monitoring, to measure the how much particulate matter or how much residues are present in the pharmaceutical manufacturing waste regulations. That also we are using this method, right? So these are the different techniques of or applications of the gravimetric analysis, right?